Hello, everybody, and welcome to episode 96 of the Hyperthesis podcast. I'm Liam.
I'm Feely.
I'm Patrick.
And I'm Matt.
As you can hear, we have a very special guest today, Matt Campea. Thanks for joining us, Matt.
Yeah, thanks for having me. Glad to be here.
So a quick introduction about Matt. Matt, our special guest today, is a person who always loved science, but he took it to the next level when he majored in chemical biology at McMaster University, which he claims that chemical biology is not the same as biochemistry, so please stop asking that. We're probably going to ask you about that later, just a heads up.
That's fair.
After Matt gained some co-op experience at a cancer research hospital and a pharmaceutical marketing agency, he started his PhD in chemical engineering at McMaster under Dr. Todd Hoare. Matt's research focused on drug-loaded nanoparticles to treat various diseases, such as cancer and schizophrenia. Specifically, he explored how nanoparticle size and shape could overcome biological barriers in each disease. We've actually talked a little bit about biological barriers before in the past. One of our guests, Hannah, was working on the blood-brain barrier. So Matt worked on biological barriers for these diseases and how to overcome them and improve efficiency of drug usage, or so forget the word right now, but how drug delivery, that's what I'm trying to think of. Nowadays, Matt's a scientific specialist with Cytiva. Is that how you say that?
Cytiva.
Yeah. Sorry, Cytiva.
It's okay.
Yeah.
Everyone likes a little giggle, and I accept it. I love it.
All right, little joke there for some people. Cytiva, where he gets so Matt works there as a scientific specialist, where he helps customers develop nanoparticles in an industrial setting. And when he's not working, he's making TikToks, and doing Instagram Reels. So you're also kind of a social media influencer in science. So that's also something I'm very interested in. Making reels, you make reels about biomedical engineering and things like that. You also are a very big sports fan, specifically soccer. The team AS Roma.
That's them.
I don't follow football very much.
If you're
soccer,
if you're going to, don't follow them. It's just disappointment and heartbreak, but we love them all for it.
They
We
want
do
to
it
see
live.
many Italian leagues fan.
I feel
yeah. Go ahead, Liam. Sorry.
That's okay. My bad. I just feel like everyone I know who follows a sports team just says, "Don't follow this team."
Sounds about right. That's how you know they're a real fan.
Yeah,
They've seen too much.
they've been there through thick and thin.
Exactly.
So thanks for joining us, Matt. But before we get to you about your work and your experiences and your past research, we have an intro topic today, which I have prepared. So I found a paper, and by me, I mean my partner, Nayeli, who also turns out works for Todd Hoare. So she's currently in the group that you left, or you graduated from, I should say. And I asked her to find me a paper that might be a good intro topic, because I had some ideas, and I think she saw them and told me they were bad ideas.
Thanks, Nayeli.
Politely, of course.
Of course, as she always does.
Yes. As a theoretical physicist, I am definitely don't know too much about drug delivery. So she gave me a very good paper titled "A Reconfigurable Reconfigurable pro-angiogenic." I can't even say that word.
Angiogenic?
Angiogenic. Pro-angiogenic hydrogel patch. All right, let me start that again. A reconfigurable
Angiogenic.
Angiogenic. There we go. A reconfigurable angiogenic hydrogel patch enabling minimally invasive drug delivery. Holy smokes, that was a mouthful for me. I apologize. So I read this paper. It was published in the American Chemical Society Journal of Applied Materials and Interfaces in August of 2024. And the paper discusses how The insertion of bioactive molecules and drugs into people can help treat injuries, diseases, and tissue defects. But often the methods they use to deliver these molecules or drugs are pretty invasive. For example, a doctor might need to create incisions into tissue, can result in potential complications like infection, scarring, or pain, amongst other things. There are other methods available for drug delivery, but at least the ones this paper is concerned with, the majority of them kind of they depend on the skill of the user or the doctor, whoever's injecting them, which means that there's always potential for risk. Um, Even the most skilled person who's putting delivering the drugs could have a mistake since it's a skill based thing. So hydrogel, something that I believe you might know a decent amount about, Matt. So please, tell me where I'm wrong and where I'm not wrong. Hydrogel is a popular material that's used to deliver bioactive molecules, and I know part of your previous supervisor's research kind of involved that a good amount.
Mhm.
So hydrogel, my basic understanding of it is it's a substance that's composed of polymer chains that are hydrophilic. So that means they can absorb and retain water or other fluids or substances, and it gives them this gel-like consistency. So this means that hydrogels can be categorized based on their response to external stimuli, such as temperature, pH, so the acidity or basicness, and that means that they can be engineered to release these specific substances that they've absorbed in kind of a controlled way in response to these stimuli. So they're very useful for the delivery of molecules. However, the paper that we're interested in today turns out that hydrogel can still suffer. They talk about how hydrogel can still suffer from these kind of previously mentioned problems when delivering the drugs. Even when you make efforts to make the gel minimally invasive, there can still be problems. I believe that what usually happens is that, at least with this hydrogel, again, maybe this is just a really specific case the paper's considering, but they made it seem that usually what happens is that you inject this kind of hydrogel substance as a liquid into some tissue using some kind of catheter, and then afterwards the substance goes inside and it gels. So it turns from a liquid into a gel once inside the tissue in the desired area. And the paper discusses how currently one method that they use to inject um, this catheter method, hydrogels have this shape memory. So one method is you inject it, and then you use the fact that the hydrogels have shape memory, which means that they can roll or unroll depending on stimuli, so they can kind of like fold and unfold themselves.
A gel is a liquid.
Depends who you ask, probably.
Gel is a liquid. It's
Is it?
Well,
I don't
it's a
know.
suspension of solids in a liquid.
Mm.
It could be.
Is it? It's
Maybe it's more than that.
maybe this is like you know how there's like 20 kinds of ice or something weird like that, right? Maybe there's like different phases of gel. I don't know. Matt, do you happen to know about that?
So actually, I'm really glad that Phil, you brought this up because that was the exact kind of, I think, point I was hoping you would make. It's just like this level of physics. I think like when you have like a physics brain, I do not have one, but you three definitely do. And I think there's like this level of understanding that you want to get so deep down. It's like, honestly, you probably thought harder about this than I have. And I thought about it for 6 years. So it was like, I have no clue. I think there's a lot of um general there's different, I guess, definitions of what people will classify gel as in the engineering space, or how do you properly quantify that. And most that I've seen assume a gel is different from a liquid. What exactly that means depends on person to person.
Okay, let me
I
put it this
know.
way. There are solid, liquid, gas, plasma, Bose-Einstein condensates. That's it for now, right? Like, where does gel belong? Is it a
Well, what do
solid
you?
though? Or is it a liquid? It's definitely a fluid.
There's more to it than that, but there's like you can have super solids, which are super fluids and crystals at the same time. Those weird things like that. But there, our previous guest, well, one of them, Hamza, he works for a guy who studies soft matter physics, and soft matter physics is kind of like this whole thing where you're in the gray area between solids and liquids and gases, mostly liquids and solids, I believe. So it's like, is Jello a liquid or a solid? It's like, well, depends what time scales you're talking about, depends what you're doing to it, and depends what kind of Jello you have. There's different kinds of gels. So I don't know the answer. I can understand that if you ask this question, you're probably going to get a bunch of different answers depending who you ask.
Yeah, I would say most common definitions err in that same space as you were just mentioning, Liam, of what the soft matter physicists would say. The terms that most hydrogel folk will use is that it has a viscoelastic behavior, so it is both viscous and elastic. So part of it just flows, and then another part of it will snap back into shape and have a little bit more of like a loose solid feel to it. And it's tuning those behaviors of both whether it's viscous or whether it's elastic in order to make a hydrogel for whatever application that you want. That's about as in depth as I can probably go with my definitions of hydrogels. I did my hydrogel work more vibes based, but in terms of like a hard definition, it seems to float in that in between area of a little bit of solid, a little bit of liquid, a little bit of viscous, a little bit of elastic.
Yeah, I guess Feely, you're probably right in that in some sense it's just a fluid from like a fluid dynamics point of view, but I know everyone in those fields, they kind of do like what you said, Matt. They're kind of more of like the vibes. The what type of gel do you have kind of depends like on its qualitative behavior a lot. Maybe there's ways to quantify it. I don't know.
At the end of the day, it doesn't really matter, right? These kind of categorization or distinctions, like practically, you don't really care, like what you name it. It matters like how you can apply it, how you can use it. Like we can discuss
Exactly.
semantics all we want, right? But it doesn't change the fact that we need to make this hydrogel into applications.
Yeah, so exactly what this paper's doing. They're not debating that kind of stuff. They're just saying how can we use this to deliver drugs in a non-invasive way. So coming back to that, the paper discusses how there's kind of like two different methods that they're interested in. Where one of them is that you inject the hydrogel into the tissue with a catheter, and then it gels on the inside, so it becomes less liquidy, I guess, and more gel-like. And what you can do is you can exploit the fact that sometimes you can make certain gels. I think you can like put them in. You can put them in like bi-layers, combine different layers of them, and make it so that they have shape memory, which means that if you expose certain stimuli to them, they roll or they unroll up and expose the drugs at kind of controlled rates. Well, another method that they talk about is to inject the gel and then kind of manually roll and unroll it after the liquid's gone inside and gelled. And I'm not exactly sure how that works, but I imagine they say that it's invasive and it's a skill based thing. So I imagine that they inject it and someone has to like kind of manually, I don't want to say use some little tweezers, but there's something they have to do. They have to go inside the incision and roll it themselves. And the former method, the kind of automatic rolling one, means that the injection is kind of limited to a specific size and structure, which kind of restricts its scope. So you can only inject certain size things. You can't really like once they're in there, you can't change them very much basically. So it's a bit more restricted, and you need to know what you're doing in advance a bit more. Whereas the latter method, with where you go in and manually fold it, that's like I said, that's a bit more invasive and is highly dependent on the skill of the operator. And there's also risk of material fracture. So you could, I don't know how gel fractures, because when I think of fracture, I think of solid objects. But basically, I think there's risk of breaking your gel into multiple pieces.
If I could just quickly actually add a little bit of context to that, when thinking of hydrogels, I think in most cases in biomedical engineering, honestly, just genuinely picture Jello, like the consistency and feeling of the Jello that you would eat. That is more or less what most hydrogels look like, because Jello is a hydrogel, more or less. And so a lot of these biomedical applications, when they talk about fracturing, think just of that as you dip your spoon into some Jello, how it just kind of breaks apart. There's a little bit of crumbling, but there's a little bit of wobbling as well. That's, I think, their fear is if you, for certain applications, if you don't have this nice intact gel, it might not perform the way that they want to, is the way that I'm interpreting this. But
Yeah, and I think
I
that's
could be wrong.
yeah, I think that's right. So what did this paper do that's different? Because they talked about kind of, I don't want to say failures, but problems in the current approaches. So this paper claims to address these issues, and the scientists and engineers developed a reconfigurable hydrogel patch that can self-fold into a small tube and unfold spontaneously once it's been implanted into the desired tissue through a catheter. So I'm not I'm not 100% sure how that's different from the first method they talked about, but I think the idea is that it can fold and unfold, whereas the original one maybe it just goes in and unfolds or something like that. Um, it wasn't super clear to me reading it, but again, that's more so probably my own fault than the paper's fault. So yeah, so they have this. They assembled a hydrogel that could achieve this goal by layering a drug releasing polyethylene glycol diacrylate hydrogel sheet. That's a mouthful. I'm not sure what that means, but it's some kind of molecule compound combination thing. Um, so they have a sheet of that, and they combine it with another sheet of polyethylene glycol diacrylate and polyethylene polyetylenimide. I don't know. Um, something else. They have one sheet of something, and then they have another sheet of the same thing with something else in it. So they've got two sheets, and one's slightly different from the
That's
other.
very
That's my
precisely. I love it.
that's me, man. That's me in a nutshell. Words are hard, so I make up easy ones.
That's why we invent symbols in physics. Like, hey,
Yeah,
it's gamma.
exactly, exactly. So they combine these two sheets together, and the sheets rapidly swell and degrade, kind of evenly throughout them at controlled rates. And the researchers are able to control these rates in the self-folding dynamics, or the self-folding and unfolding dynamics, by controlling differences in the expansion ratio and uh, elastic moduli between the two different sheets. Um, and maybe for clarification, I can't remember if I said this or not, but the unfolding is what triggers the sustained release of the drugs. So that's what you want to get it in there, and then you want to unfold it. And these researchers, they believe that this kind of gel bio patch will have broad use and for minimally invasive delivery of a wide array of therapeutic molecules. So, like I said, it wasn't 100% clear to me the difference between the first method they mentioned and the second one where you have to go in and manually fold it or unfold it or fold it. I can see that that's quite invasive. But the first one where they said you inject it with a catheter, I'm not sure if they had some other different method that they used or some mechanism that maybe isn't as useful, or maybe it only unfolds a certain way and you can't control it. But at least this way seems quite useful because you can really control what's going on.
I'm very curious in this use case for kind of the automatic unfolding mechanism, if that's kind of an all at once type of thing or if that's over time and what that distribution looks like. Because I know for some drugs you want to deliver them over a longer period of time. So does that mean just separate injections because it's all happening at once? Or is it controlled in like almost like a normal distribution or a controlled distribution where when they hit whatever they're interacting with in your body, if there's like slow controlled openings of some but not all, so it's kind of this not necessarily random process but a slower process.
I think that's something they can control. So I don't know if they can just unload it all instantly, but they have all these plots that I'm looking at right now in the paper, and they're showing like different plots over time. So like how the radius of unfolding, or the um what's this one? So I have to turn my head sideways to see it. They have the radius, they have expansion ratios, inelastic moduli, and somewhere I thought they had a concentration one. But basically, the kind of time scale over which they're um, measure or taking these experimental measurements are over like minutes. So anywhere from like 10 to 90 minutes, roughly. So I think it comes out slowly, but not super slowly. Like I don't know if you could leave it in there for a week and have it still releasing stuff after then. So it seems like it's slow, but not super slow. That's what I'll say.
Well, I think we should move on because I think I am more confused than when you started. It's like, what's going on with this, man? Like, I have no idea what you're talking about. There's bilayer, there's hydrogel, and expansion. So, let, let's keep going.
Well, I don't know if that made sense to you, Matt, but
And shockingly, that made a decent amount of sense, Liam. I thought you did a great job.
thank you.
If we can move on, happy to move on. If we'd like to
Yeah.
do a quick summary, can also do that. Whatever you're in the mood for.
No, I think that's all right. I thought it was interesting because I'm so used to these theory papers, right? And then you get a paper with I think they have two equations in it, and they're like the simplest equations you've ever seen.
Yeah,
Which
that's
is
two more than most, I think.
yeah, I was gonna say most maybe have like one, maybe. And then don't really they don't cite where the equation comes from, so it must be pretty standard in the field. It's just like the radius of unfurling, I guess. So they did match their experimental data to some theory, and it matches pretty well. So that's always good. Anyway, that was the intro topic. Some hydrogel drug delivery, so kind of relate it to what you might have done in the past with hydrogels and now and drug delivery. I'm not sure if you ever used hydrogel for drug delivery or not, but we'll ask you about that now. Well, in a minute. So before we get to your PhD stuff, can you tell us, Matt, what you're working on now? Or
Yeah,
like
happily.
who you're working for this company,
Mhm.
doing as a scientific specialist. So what does that even mean?
That is a great question. And my full title is actually Scientific Inside Sales Specialist. So it's like a branch of sales, but it's like in the very specific scenario of when people will go to our website, which is a website that has a number of different instruments for biotech and biomedical engineering. And if they're interested in learning more about an instrument or they want to, potentially buy one and they hit the contact us button, that comes to me. And then basically I teach them about our instruments, how they work, and how they might be able to use it for their very specific project. Sorry. And a lot of these projects, a lot of the people that we work with are people who are trying to make nanoparticle based drugs. One classic example would be the COVID vaccine, right? Like that is a nanoparticle. It's a vaccine that is delivered as a nanoparticle, and our instruments make those nanoparticles. And I basically talk to people about nanoparticles all day, and it's pretty sweet.
I think the burning question is, what is the difference then between the biochemistry and the chemical biology?
Great question, Feely. So there it all stems from there's two distinct programs at McMaster University, which is where I did my undergrad and fun enough my PhD. But there is a biochemistry program and there was a chemical biology program. And honestly, most people don't know the difference until you are actually in it. But basically, biochemistry is very protein and much more biology heavy. And then chemical biology is basically organic chemistry, but you see how it applies in the body. So that is both, making small molecule drugs and whatnot, or seeing how certain interactions in your body, like what is the chemistry behind that interaction, rather than protein A attacks protein B, and then that leads to protein C or whatever, because that's what goes on in biochemistry. Apparently, is a lot of these like protein pathways and what's being increased and decreased. Whereas chemical biology is more like what is the chemical group that is interacting with another chemical group that causes that biological interaction to happen.
I feel like they should switch names just because that's not literally the opposite is what my intuition went. It's like, oh yeah,
Really?
biochemistry is the chemistry of biology stuff, and then the what's it called chemical biology is the opposite because the
Yeah.
English use adjective first, right? To emphasize the noun after. So it's like so when you say biochemistry, it should be chemistry mainly, and then bio second.
My working theory is that biochemistry came first, and the chemical biologists like, okay, wait, what do we do? And so they just said, well, we're going to be chemical biology now, and they kind of ran with it. Proven by absolutely nothing. Like, there's absolutely no data or proof behind that. That's just a good old Matt theory.
I can believe it.
So in
Thanks, Liam.
in these these fields like either biochemistry or chemical biology, when you study them, are they like very specific to cases? Like when you let's say you are learning to be a doctor, they usually study specific diseases and stuff, right? I wonder how theoretical these fields are because in physics we kind of like we don't really engineers study specific machines and engines and such, but physics we look into more like fundamental nature or like how to describe things generally. So when you take these fields, like what you do and stuff, are they very specific or very like theoretically general?
I think how specific you are, how general you are, again, it super depends, right? Like I know many physicists who they've spent 8 years studying one crystal, and that one crystal has does things that no other crystal does, and that's what they're studying. It's very like,
Oh,
you
that
know, it
those are
really.
experimentalists that do that though, because for theorists, I think
Mm,
even though we care for one crystal, we want to generalize that behavior to let's say a group of crystals, a family of crystals.
yeah, fair enough.
Yeah, I think that's a really good point. Feel actually, both of you had very good points. I was actually originally going to give basically the same answer as Liam, which is like, oh, it kind of depends. But um, I think you end up seeing more general. I mean, there is obviously that range, but chemical biology, because it's a little bit more of an applied field, it's not like theoretical chemistry, or you are all more familiar with of theoretical physics, where to your point, you're just trying to prove certain things are working from a fundamental level. There is this level, I guess, of applied. Like chemical biology is a bit more of an applied field, and so I think you're always going to have a little bit more general, in my opinion, just right out the gate because you're trying to solve problems, and you might say, "Well, this is my one solution. It's kind of specific, but we're really trying to solve this general problem or answer this general question." Such as, how can we treat, I don't know, Alzheimer's disease, for example, would be, developing a drug that would treat Alzheimer's disease. And you might have one strategy for it, but you still need to know the other specific strategies in order to solve that. I don't know if that answered your question, but um, that's like a high level overview, I would say.
I'm wondering, coming back to your current job a bit, what's kind of like your experience working with customers, I guess? Because like, when I think of customers, I think of like I go to the store and I buy something, but it's a bit different when in your field for sure. And when you get to these kind of things where you're helping them like develop nanoparticles, right, for industrial settings, I can imagine that's kind of like the people you deal with. Are they like more business oriented, or were they also chemists or engineers, or how does
Yeah.
that work?
Most of the people that I talk to are either currently in grad school or have a PhD. Some of them are master's students, but for the most part, it's people with PhDs. It's people who are trying to solve a certain scientific problem or a certain engineering problem, and they need help to do that. So it's usually a very technical conversation. At some point, either it will delve into business or there will be a business aspect, but it's usually, if not always, science first. So I think it's a great example that you bring up, Liam, of this grocery store analogy. Or even when I was going into it, I was always picturing like, well, if I had to buy a car or something, and you're just thinking of in this car example, I would be the sleazy car salesman trying to sell you a van with three wheels, but that's not the interaction at all. It's like you have to know your stuff, and you can't be that sleazy salesman because they'll call you on it. They know they're smart. They'll call you on it. So
And the other thing too is, I guess, if you're doing like, medical applications, I don't know if that's because you said industrial settings, but if you're doing medical applications, like you can't
Mhm.
really, half-ass something like that because it'll
No,
have big
not at all.
consequences. And there's a
Yeah.
lot of strict approvals and a whole process to go through
Mhm.
depending on where you are.
Yeah.
So these solutions you have in sales and stuff, are they like equipment or like are you selling the methods? Like I'm not sure what exactly is on being on sale here. Like because like but let's say like we have a company, not we, like in physics people do like optics. So there are companies that sell like advanced optical equipment. They're very expensive, but they're very highly precise and they're very specific. Like it's like the same thing where they have like customer representative or sales guys that know their stuff when they talk to the lab. Like, hey, we need exactly these wavelengths and all this filter, this dispersion, and you really need to know the stuff. But they really just sells you the like not the method, right? Just like here's the lens that does this. But during the consultation and such, it has to go to a lot of details. With like, are you guys like offering solutions or mostly just equipment?
Yes, we actually do both. So we actually do solutions, both theoretical and practical, and we sell instruments as well. And I think the easier example that to grasp is here is an instrument, go ahead, and then other things that go with the instrument. So certain like chemicals and stuff that we know are compatible with the instrument, and we know that people can, someone with 2 brain cells can use these chemicals and these kits in order to make a very good like pharmaceutical product, at least from from certain engineering standards. But we do also offer these services where if someone needs some more knowledge on what we call nanomedicines, which is just medicines in a nanoparticle form, or hey, sometimes people will ask us to make nanoparticles for them, and then they will basically take them back afterwards, and they'll see if it works in their very specific biological application. So there is this idea of doing both, which I think is really really interesting, and it's something that we obviously like a lot. And we very much pride ourselves on, having just a whole bunch of nanoparticle nerds, myself included, just kind of waiting in the wings, waiting to solve different technical challenges that come with whatever medicine people are trying to develop.
Do you have do you have customers from all over, or is it more like localized? Because again, we're always going to talk about differences in physics and chemistry because we're all physicists. But like what Feely was saying, these optical people who have all these fancy instruments and lasers and mirrors and things like that. Like I've met some of them and talked to them, and they ship things all over the world. And I guess there's a bit of science to shipping these fancy optical equipment because they can get bumped and damaged. And I don't. It's probably different with chemicals and nanoparticles because I don't know if some of them have lifetimes or
They
things
do.
like that to consider.
Yeah, they very much do. And I think a good example of that is even, again, bring it back to the pandemic where, people knew that we had to ship these vaccines all over the world, right? And they typically had to be shipped in very, very, very cold temperatures that it makes shipping an absolute nightmare, right? Like no one wants to ship their things where it has to be -80 all the time, like -80 degrees Celsius all the time, because it's a lot of effort and it's a lot of money. But that is a big part of it is when you're trying to ship, say, to a clinical trial site, which, sometimes we do, you have to make sure that when you're shipping it, it is arriving in the same state that it left in, and making sure that once it has arrived and it has been whatever it is, unthawed or sorry, thawed, or unfrozen in any way, that it will then work in a person in the same way that it worked when you had one fresh in your little facility when you made these medicines, right? So yes, there's definitely like a big science and whatnot to just shipping. Luckily, that is not my job, and that is the kind of the beauty of being in a big company is that is someone else's job, and they are good at it. I wouldn't know the first thing about it, but they certainly do.
So then I think I want to pick your brain on more your technical expertise then, apart from all the sales stuff, your company. I think a lot of people wonder, how does drug delivery work? What are the barriers? Like, you mentioned the drug delivery barrier. What does that even mean? In physics, we have energy barriers, potential barriers, and they're very
Right.
abstract. But it seems like in these type of like medical research, what do you mean by these barriers? Are they like, the phospholipid bilayers of the cell? Are they like blood? Are they like
That's one of them.
active
That's another one.
transport problem, osmotic
Oh,
pressure? Like, like
you're
what do you
listing
mean?
all of them. No, you just listed a bunch of the different barriers. Is pretty much anything that your drug or your medicine comes into contact with could be a barrier. What is something that is preventing your medicine from working is something you have to consider when making or delivering a medicine, right? So I think the there's 2 examples I think I want to give. One is the easier one that I think most people will understand, which is imagine you have a brain tumor. How do you get a drug there? How do you get a drug to try and treat that brain tumor? There's a couple different ways. You could try and inject something straight into someone's brain, or you could try and give it to them in their blood. and trying to allow that medicine to get to the brain eventually, right? Because your blood circulates all the time, and blood is obviously getting to your brain in some capacity. But is every content of your blood, including this medicine, is that getting into your brain? Well, we don't know. So we're going to try and design something that will allow it to get to the brain. The other example would be treating like a skin rash, right? Like trying to deliver a medicine to your skin is a little bit easier because you rub an ointment or whatever on your skin. You are physically rubbing that medicine into your skin. You wouldn't want to inject a medicine into someone for their skin when you could just do it right away. So it's taking each of those challenges and taking each of those potential barriers. In case one, it is trying to get to the brain, or as you mentioned in a previous podcast, you discussed the blood-brain barrier, which is that thick layer of cells, basically, and other components that makes it very specific as to what actually gets into the brain. So crossing that blood-brain barrier is a much different biological challenge than, say, treating a little skin rash where you can see it and you can touch it and you can treat it right away.
Let me be more specific, because maybe I'm not really understand the term delivery too, because like, what do you mean delivery? Is it mean going inside the cell, or does it mean just be in the fluid surrounding the cell? Like, or are they specific to, let's say, your targets? Like, what does it mean to be delivered?
Yeah, that's those really, really good question. I think especially because this is a very hot button issue in the field of delivery is what does that even mean? And it really is all of the above. And I know that's not a great answer right away, but I'll explain. Is trying to take a current medication and just putting it into a nano-sized material, so into a very, very small material. And can it do all of those things? Can it get into the space around what your target tissue is, or into that target tissue, but also specific cells within that tissue? If we think of, literally any tissue, we could talk about, let's stick with the brain example for now. Different parts of the brain have slightly different physical features, maybe slightly different cells or slightly different, biological components, if you will. And how do we get our medicine? What if our medicine only works in one of those areas of the brain? How do we get that medicine to that very specific part of the brain? Is really what we're trying to answer with all drug delivery issues. And then just, change brain with any target organ. So, whatever, lungs, liver, heart, cancer is a big one. That's what I did most of my studying in is, how do you get to cancer, but not try and attack the healthy cells and the healthy tissue that is around it, et cetera, et cetera.
So how do you actually go about testing this? Because you can't just jump right to clinical trials where you're like, oh, just try this, see if it works kind of thing. How do you actually, I guess, recreate those barriers, whether it's through like human stand-ins or if it's completely artificial? And then I guess, how do you even prepare for that? Just is based on previous research of, oh yeah, this is able to like penetrate the blood-brain barrier, whereas this isn't? Or is there, I guess, a theoretical approach behind it too, where you're saying, okay, well, a molecule of this size could probably go through and testing it like that? So what's the approach to actually testing all of this?
Yeah, it's a really, really good, really, really good question, Patrick. And it obviously starts, I think, like most, I think, wet lab research, it starts with a theoretical of some people wrote some paper that said that a molecule or a particle of a certain size can get through a certain barrier. And then you go in and you try and test that in a slightly different application. Now, in terms of the general progress of what that would look like, obviously it would, you make your material and you see, is this even good in the cells? Like, does it even help the cells that you're trying to help, or kill the cells that you're trying to kill? So for example, like in cancer, the first thing you want to do is test, does your nanoparticle, does it actually kill cancer? When it's just exposed to nanoparticle plus cancer, do we kill cancer? Yes or no? And then from there, some people will do other types of like intermediate experiments. So sometimes there are some, you can make artificial barriers depending on what that application is. That's a little in the weeds. I don't know if I can do the explanation justice right now without throwing out more just random examples. But the next step is typically animals. So it'd usually be go cells, then some kind of like artificial amount, sometimes cells in that, and then it's small animals like mice and rats, then slightly bigger animals, which might be, dogs, cats, rabbits, and non-human primates. And then finally, then you're kind of at this stage of, oh, now we can try it in humans because we've seen there's been enough steps along the way to show that it should help.
I
If
see.
that makes
So in this type of research, what is this? Why nano? Why nanoscale? What's the significance of nanoscale? Well, in a little bit of context, because the water is like water molecule is like a fifth of the nanometer,
Mhm.
right? So why is it so important that we work in the nanoscale in this type of delivery?
Yeah, nano seems to be that kind of sweet spot between targeting certain parts of these barriers and not just getting into absolutely everything. So if we think of something like chemotherapy, that's just kind of where I did most of my research in, was trying to use chemotherapy or trying to make chemotherapy better. So targeting cancer. So most people will know that chemotherapy is very non-specific. So it's small. Most chemotherapies are small molecules, so they're small enough to basically get into any tissue, any cell, and it ends up killing that cell, whether it is cancer or it is healthy. It's very non-specific. So what people have found is that if you then take that same very small molecule, which is usually on like the picometer or slightly like less than a nanometer scale, but you put it into something that is on the nanometer scale, it doesn't go into every single cell. It will go into maybe a couple healthy cells by accident, but it will mostly localize into tumors. And this is where, at least from my research, where you see the biggest push was people saw that it was really good in cancer. Like nanoparticle based drugs were really good for getting into cancers and not getting into your healthy cells. And then people since then, over the last maybe 20 or so years, have been 20, 30 years, have really been trying for this push into, okay, well, what other organs and what other applications does this nanoscale work in?
Yeah, just even like talking to Nayeli and her coworkers and stuff, it's like it feels like you can really target like you literally just said, right? Like you can target very specific things with nanoparticles without worrying about much else. I mean, obviously there's a lot of stuff that goes into that, but very useful.
But yeah, exactly.
I am curious about your thoughts of, I guess, drug delivery, or even, especially in the case of cancer, specialized treatments. Because I know there's the side of the nanoparticle and drug delivery where it's targeting, say, specific types of cancer cells. But there's also a whole other region of research, I guess, coming more so from the biology side of like using your differences in immune systems and kind of using the human's biology to target that. I'm just curious about your thoughts on that and those differences.
Anything in particular, like any particular differences that maybe you've read that, you want me to elaborate on, or that sparked this question?
Well, I mean, I guess uh, your research has kind of come from the chemical side of, okay, what can we manufacture and put into the body of uh, drug delivery, whether it's for cancers or let's say Alzheimer's or things like that. Whereas there's a lot of research, especially in microbiology, where we can um, either use viruses, for example, as instead of producing nanoparticles and filling them with drugs, we instead create kind of viruses where they're able to be specifically encoded to interact with, say, the outer layer of cancer cells, but not other cells. So just different method of delivery, but kind of more, I guess, from the living side of things, if that's a fair way to describe it, rather than the, I guess, inorganic, or not even inorganic,
Like the
but the
synthetic, I guess.
synthetic side of things. Yeah.
Is a virus alive? Is
Not
it a
outside the
computer?
host.
Yeah, it is not. I'm not a virologist, but from what I can understand, they're not alive. I'm pretty sure. I really
Yeah,
hope.
I think that's
I
right.
really hope a virologist doesn't come in here and be like, well, actually, but I'm 99% sure they're not fully alive, because just like you're saying, Feely, they need something to grow with. Otherwise they just kind of sit there and they just have their own little genetic material. But to answer your question, Patrick, I think I kind of am agnostic to what is. I don't really have a dog in the fight. Like, that's not true. I'll ride or die with my nanoparticle boys, but it'd have to make sense. I think with any kind of treatment, it just has to make sense. So this is something we actually talk about a lot, especially in the nanoparticle world. They got very popular because of both cancer and then infectious diseases, where typically you were using virus-based vaccines to, you know, do vaccine work, do whatever it is that you want to do with that particular vaccine. And what we see is that a big benefit of nanoparticles over using viruses for therapeutic applications is that nanoparticles are much cheaper to make and much, much easier to make than viruses. So viruses are very cool because you can, they just kind of make themselves in the right environment, but it takes a lot of needing to set up these big bioreactors, these large scale reactors where you have like a lot of these cells growing, and then the cells produce these certain viruses, and you have to make sure that the cells are producing viruses with the right amount of things. And it's just like very time consuming and very expensive to set up. Whereas a lot of nanoparticles, especially in the vaccine space, it is quite literally you mix things together. You take things, you put them in some kind of solution, in some kind of suspension, and you mix them together. You might have to do a little bit of like downstream processing to fine tune what that actual particle looks like, but it's actually a simple one. It is incredibly easy these days to make them, and to make them on a large scale. Like you can get hundreds and hundreds of liters of usable nanoparticles that are ready to go in one work day. Whereas trying to make viruses, it takes like many, many days, if not weeks, to have this big scale that are ready for like human intervention.
I see. I think that's very interesting how you said it's kind of easy to make, because I study nanocrystals and how nanocrystal form. That's literally my research for the past few years. So I'm curious on so let's say I want to design a particle to do to break certain barrier, to deliver certain place. What would be the process of designing that? What do you okay, I'm looking at this scale, so I need this, but how do you come up with a shape?
Can I add on to that question and maybe make it more difficult? But can you talk about it in the context of your schizophrenia research? Because you mentioned that you also studied schizophrenia, not just cancer, during your PhD.
I knew I shouldn't have put that in there. I'm not the schizophrenia guy, but I'll try to do my best.
Okay. You can just answer Feely's question then.
No, but it was in my thesis, so that's okay. It's I need to defend it. You know what I mean?
Fair game.
We love defenses around here. So sorry, Feely. So to answer your question first, or to start with answer that question of how do you go about creating and designing one of these nanoparticles, that's really where digging into previous research really comes in hand, because you really, at least the way that we did it in the horror lab, was trying to first think of, okay, what is your biological target and what are those barriers that come with getting to that target? And that's where you kind of start digging into, okay, well, for example, if we're trying to get to the brain, Liam, as you're mentioning, as we're trying to get to the brain to treat schizophrenia, for example, the main ways, there's what I would say are there's three main ways to get to the brain, for example, which again, you get this from just reading a bunch of papers and having people tell you what are the best ways to do it. One is injecting something in and trying to get through the blood-brain barrier, which we talked about a little bit already. Two is injecting it straight into someone's head, which we also kind of talked about. And I don't know about you guys, but that stresses me out just thinking about it, just having someone do that. But and then three is actually going through your nose. So there's actually a connection where for any Simpsons fans out there, there's an episode where Homer has a crayon stuck in his brain and it keeps touching his brain, and then he removes it, and then he's super smart. That's because there is actually a nose to brain connection that allows, especially nanoparticles and nanoparticle based drugs, to get into the brain, but you don't have to go through the blood brain barrier. So once you kind of know these three methods, it's then, okay, if I choose option A, which is injecting, option B, which is straight into the brain, or option C, which is going through your nose, what are the barriers along the way? In the blood, it is making sure that it gets through the blood and then through the blood-brain barrier. Into someone's skull is getting through someone's skull. And then through your nose is actually just trying to deal with the mucus. So trying to, there's obviously lots of mucus in people's nose, and that will slow how much nanoparticles can get to the brain. So how do you get through the mucus without, going through the mucus and then swallowing some or getting it into your lungs, for example? And so you kind of just do a lot of reading and trying to figure out, well, what have other people done in each of these avenues, and which avenue do you want to try and tackle or improve on?
I will just say for a quick PSA for the third avenue through the nose is don't go diving headfirst into certain bodies of water, especially thermal lakes or thermal pools, because they have brain-eating amoeba, and that's their delivery method. They're able to
Really?
pass that blood or that through that nose-brain barrier quite easily, and you die eventually. So if you've been to thermal pools, especially there's a famous one in New Zealand, or there's a few in Costa Rica that I know about, don't put your head underwater.
Very good to know.
Oh, I learned about that nose brain thing from watching a documentary on ancient Egyptians. That when they mummify the pharaoh, they take the hook in
Right.
and take out the brain from that. It's kind of crazy.
Right. Yeah.
Yeah, that's what I was. That reminded me of that because I learned about that at one point in my life. That's kind of wild how you can get to the brain, like kind of unrelated to everything you just said, like about the drug delivery. It's just kind of wild that you can get to the brain through the nose. Like, is there any kind of barrier? Like, there's like some barrier, obviously, right? But it like allows things to pass through it.
Much easier from what I
Yeah,
can do. If it, as I'm sure we've all done in our research times, is, you get so hyper focused on, or so focused on certain concepts around the general concept, that it's like there probably is, I'm sure there's something that stops you from, it's not immediately just like nose and then brain immediately, right?
yeah,
There's got to be part of your skull, or I don't know, some kind of membrane of something or rather, but it's definitely easier than the blood-brain barrier. And there are certain nerve pathways, at least when we're talking about nanoparticle based medicine, there's certain nerve pathways that we know based on the size of those nerves and the openings to those nerves, where nanoparticles are very efficient at then getting straight into the brain.
Okay, I had to look this up just because it's interesting. So it's essentially perforations within like your sinus cavity leading into the brain that lets your olfactory nerves through. So when you're smelling, that's up in your sinuses, so you're not actually smelling in your nose, and those olfactory nerves need to connect to the brain somehow, and so there has to be channels, and then those channels are that connection. And so, of course, it's this kind of porous material that not only opens it up for nerves to pass through, but other things too. Creepy.
That is quite creepy, and it's actually kind of helpful because even though I defended my PhD last year in 2023, I am still writing some of the papers, and one of them is actually the schizophrenia project, where we've kind of just been assuming that it is just, oh, the nanoparticles get into that olfactory nerve, or some part of the nanoparticles do, and it just goes from nerve into brain. But the fact that it could also be, not actually getting into the nerve, but it sounds like there's these little holes almost around the nerve that like the nerves kind of fit into. And I wonder if that's another avenue even that we could explore. I don't know if someone else has, or if I'm totally misinterpreting what's going on here, but
thank Patrick in your acknowledgments if you figured that
oh,
out.
I will.
Yeah,
And the whole Hyperthesis podcast. Thanks.
oh, incredible.
So one of the reasons I'm interested in your design process is that I do my group does inverse design of a lot of stuff, right? Instead of figuring out the properties of particles or crystals, we do like, oh, give us the properties, we will figure out the crystal, or figure out you give us a crystal, we figure out the optimal shape that create that crystals, and we work in the nanoscale too. So our method is called the digital alchemy, where imagine, because when you make your particle in a lab, right, it's fixed. You made it, nanoparticle. You can't change it, or like it's difficult to change it. But on computers, there's nothing that's stopping me from changing the shape of the particle as I simulate my simulations, right? So we take take advantage of that and try to find like what's the optimal shape for certain properties. And I don't know if in this type of field, if there are any inverse design that's happening, because like I said that like it's so easy, just change the shape, but mathematically we have to do things to be consistent with the law of physics, right? So I don't know if you know that people are people trying to inverse design, because if you can do that, it's kind of it's like the solution to almost everything. Like, oh yeah, you want this to pass this one, we just inverse design. We need this shape of the particle and done.
Yeah, that's something that a lot of people are trying to do, but it's, from at least my experience, shockingly infant still. Like it's still very much in its infancy. Because the field currently is a lot of wet lab people, so people who want to just make these particles. And so, similar to what I was doing, it kind of goes, you make a bunch of them and then see which ones work, and you just keep going, and you keep going, and you keep trying the ones that are working, and then if they stop working, you stop using those and use something else. So there's more and more trying to decide, okay, well, what is the best way to reverse engineer these? I think the problem is there's not enough of a strong like biological correlation with certain targets and what the design of that nanoparticle is just yet. So, for example, we do talk a lot about targeted treatments. And I know we've talked about even today a little bit of, oh, you can technically make a nanoparticle that goes wherever you want. And it's like, well, yes, but there's not enough consistent, like there's not enough of I guess like an industry standard. What I'm looking for here, like it's not like a rule of, oh, if you have a particle of this size with this shape and with these materials, it will definitely get to the brain, but it will never get to the liver. As of right now, we kind of know that all nanoparticles get to the liver in like a significant
So
amount. Like
you don't have the defined parameters of what would make
not yet.
things go where. I think that's why it's sometimes the heuristic process is faster
Exactly.
because if you try to make an understanding, well, maybe there are 50 hidden parameters that you need to find to like, well, and perhaps knocking down the door is faster than find a way to beautifully open the lock, right?
Yes, I love it. Yeah, that's exactly it. That is exactly it, and that is luckily that is the thing that everyone talks about these days. Like even in my own job, I'm getting this wide range of experiences and these people who either have, they're either nanoparticle experts or they have never worked with nanoparticles a day in their life. And even a lot of the people who are very, very new to the field, new to making nanomedicines, even they're asking about targeting. They're asking, hey, well, how do we know that it's going to get this thing? So it's at least at that level where it's, if you're in the field, you have some idea of what targeting needs to be, which is much better than it was, say, 5-10 years ago. So I think we're kind of in this point where, because everyone's focused on it, there will be more and more, at least in my opinion, there will be more and more research learning how do we design these, how do we reverse engineer these, similar to what your lab is doing, Feely, for nanomedicines. It's just not there quite yet.
Yeah, I think what I find that too, that my group kind of see that, yeah, these are in its infancy. It's nobody really knows how to properly do this. I think we don't even understand how, let's say, how shape form crystals, and there are symmetry arguments that mathematics can make, but really it doesn't really work that well for like complicated crystals. And you can see emergent symmetry that comes from like, oh, your shape, your particles don't have this symmetry, but your crystal does, right? Like, so what does it mean? Like, perhaps like there's something to learn here. And at the end of the day, when we get to like the chemists or experimental physicists who make these particles, like there are so much to learn in terms like how to synthesize them that like maybe leave the job of like figuring this out to the theorists because there's so much to do into experiments because like even though there are technically finite combinations of what it could be, this is still like the numbers combinatorically large. And so I don't know, like where do you think we are now in terms of like our human progress? Have we figured out, let's say, 10% of the like biological nanoparticles? Like where do you think we are?
Oh, it's way less than 10%, that's for sure. I think we're kind of in this place. I don't know if I could put a number to it exactly, but it's definitely, we're at least in the place where, if we're looking at like the classic like Dunning-Kruger slope or whatever that is, of the more you know something, the more you realize you don't know it. Of we're definitely over that hump of, oh man, nanoparticles will work for everything. Like we are well past that. We're now, okay, there are very specific scenarios where certain nanoparticle based medicines will work. What applications can we then take those positives and draw them into new applications or new parts of the body that we haven't tested yet before? And I think that's kind of where it's at.
Do people care more about on like what the nanoparticles are, or like how to manufacture them, or like the method of delivery?
Uh, they care mostly about does it work? At least in when it comes to biomedical engineering, right? Like that is, and this is coming from having been in an engineering lab. Engineers, I think very stereotypically, in a good way, I would say, they just want it to work. And so nerds like myself care a lot about what does the particle look like and how did you make it look like that. But most people kind of just want to see, well, is it actually doing its job?
Yeah, I think the further you get from theoretical physics and the more you get to like money and applications, it's like
Yep.
who cares if I can, who cares about what's going on deep down? Like if it works and no one like if it targets the area, does the job and doesn't have any bad side effects, then it's like who cares? It works.
It's a black box problem, right? Like, yeah, here's
Yeah.
a black box to solve your problem. And don't ask. That's what happened with AI, right?
Mhm.
I mean, we currently have anesthesia, so it's like, ooh, it works. What else does it do? We don't actually know how it works, so I don't know. Weird things happen when you assume a black box.
At one point in my life, I heard someone say that we don't even fully understand how Tylenol works, and I've never fact checked that. Or like we know what it does, we know it goes and and does its job, and maybe we have a rough understanding of how it works. But I've heard people say we don't fully understand how it targets certain things in your brain and your body and stuff. So yeah,
Interesting.
I don't know. We know a lot about many things now, but we learned quite quickly. I feel like that we don't know a lot of stuff about many things. You still see papers come out that are like answering questions that you might think are super basic, but are very deep questions.
Yeah, I think always there's that aspect of it, but I think on the Tylenol point of view, that was actually I don't know how much we know about why it's targeting certain parts, but one of the first things in my I can't remember if it was my second year or third year, this like one of our chem bio classes, what it was actually what is the exact binding pocket of Tylenol or whatever acetyl salicylic acid to certain receptors that it's to certain proteins in certain brain cells, and it like he actually mapped it out. And he's like, here's a little drawing of what the molecule looks like. Okay, here are all the different amino acids and what their chemical groups are, their functional groups, and this is the pocket, and this is why it works so well. Is because it's um because it has like a strong chemical interactions within that binding pocket. But you might be right. Like we don't know. Okay, well, how is it getting there and not everywhere else? Is it getting everywhere else in the brain? It just you don't notice the effect. Is it getting into other parts of your body? We just again don't notice the effect. So
Yeah, and I also could be wrong saying that. Like I heard this from someone and I never bothered fact check it.
one
So
of those.
yeah,
Yeah.
citation needed, right?
Yeah. So quick note to Tylenol, it's just a trademark. The drug is like
Yeah.
paracetamol. Yeah, but so I want to pivot into your life a bit. So you finished your PhD and such. What got you into this, your field, this kind of medical, chemistry, biology stuff? Like, what got you interested in this field? Like enough to do PhD, which is a long commitment.
Yeah, I was one of the classic grad students who's like, or one of those classic students like, I'm never gonna do grad school, and then you keep going. You're like, okay, well maybe, and then it's like, okay, but I'm gonna start a master's. I would never do a PhD. And then you get into the master's, and you're like, okay, but maybe it's kind of interesting. So I think for me, it's always just a general interest thing. I think I've always been interested in chemistry, but I always was also interested in biology. And at first, I liked biology a lot more. And then the more I did biology, the more I did not like it. So I was like, oh, I actually like chemistry a lot more. How can we blend these two things together? And I think I always was very interested in, okay, why are certain pharmaceutical drugs working the way that they're working? Like, what is causing that? And um, kind of going from there. I didn't know what nanoparticles were until I interviewed for a PhD position. I had some background into the field of drug delivery and like biomedical engineering as a whole through certain classes and through my undergraduate thesis. I was kind of introduced to that, but I didn't actually know why it was interesting until I started doing it. And then once I started doing it, it was all I wanted to do. And so I just kind of kept going with it into a PhD.
Okay, I think I have a maybe not similar, but I want to hear if you agree with this. So when I did my let's say when we are young, we learn mathematics when in grade school, when you get to middle school. But like you learn how to add sub and subtract, and then you learn to multiply. And then, but you can't really. It's hard to learn multiply when you don't know how to add. But once you learn multiply, you're like, adding is easy, right? Like, and then you learn calculus. You're like, oh yeah, but if you don't know functions and all these stuff, and now calculus was felt really difficult. But then you do advanced calculus, like, oh, calculus is basic stuff. So for me in physics, the reason I go to grad school is that undergrad provide us like very basic mathematical tools to understand nature, very basic. And you get to grad school, it gives us much more rigorous mathematical tools. And I thought, oh, I knew grad school math now. I can understand a lot of physics. Well, it turned out the grad school and PhD is just a starting point to be able to understand let's say the current literature in terms of science. Is that true in bio and chemistry too? Like you have to know like basic organic chemistry, which when you study it in undergrad felt really difficult sometimes, right? And it's like maybe this is the hardest thing. But when you get to grad school, oh well, that's just like learning how to add and multiply when we were young.
Yeah, I totally agree, and I think that is very much what it is. I think that's. I mean, maybe you can all speak to this as well. Is that not just like the common? I think that's the common grad student of it all, right? As you learn one thing, like, oh, that was really cool. He's like, but why? And then you just keep going into these rabbit holes of, well, how does that work, and how does that work? And then 2 hours and a billion YouTube videos later, you're like, oh, so that's how this works. And the first question you had at the beginning was actually a pretty straightforward question that you have since answered.
In physics, a lot of the time you realize that people just don't know. You're like, oh, why is, how does this work? And then it's like, well, on the deepest level, we don't really know. We just know it does. And that's kind of how I got into physics instead of chemistry, because in high school chemistry, they teach you all these rules and the periodic table and all these quantum numbers. And I remember being like, where do all these quantum numbers come from? What are they? And then the teacher was basically like, well, you're going to have to go do or not a PhD, but like a bachelor in physics to get that answer, because you have to go solve the Schrodinger equation and quantum mechanics, and all those quantum numbers kind of nicely drop out.
I think like and kind of question to you, because I feel like my field of study is a little bit easier to explain fundamentally because it deals with biology. It deals with people's body. Like everybody is aware that they are in a body, and so you can apply any of the learnings that I have and simplify it to a point of connecting it to people's bodies. But how do you do this with theoretical physics, right? Like you're talking about, like Feely, when you mentioned crystal structures, like in chemistry we learn a little bit about that. I'm also a fake like chemical biology is a fake chemistry, so we didn't really do a whole lot of the crystal stuff, at least not to this level that you're doing it with. But how do you explain to someone like, well, this is what a nanocrystal is, and this is how it forms and why it matters, but how do you minimize the amount of just everything going over people's heads?
I don't know about you, Feely, but I use a lot of analogies. That's kind of my strategies. I'm like, think of a car driving down a street, like that kind of stuff. Or think of a ball and another ball, and they're talking to each other. Like just really, really simple analogies, because um, I find it's easy to make simple analogies in at least like theoretical physics, because theoretical physics is that you're trying to ignore everything that's complicated and just break things down into really, really simple terms, I guess. I mean, easier said than done, right? But that's my thought process.
I think there are two parts to that answer. First, you have to break it down because first is what Liam talked about. Explain how it works. Great. It used analogies, whatnot. But the more important, the much more difficult part is explain why they should care.
Yes.
It's much harder because like now I could talk about crystals and why nanocrystals is important and blah blah blah. But what I research is very fundamental in a sense that like it might take years for this to be able to apply at all. So is in a sense like we have to relate to what you do. It's like, well, it kind of lay foundations for understanding, let's say, hey, maybe it would make us understand drug delivery better if you understand how nanoparticle form, or how would it actually, what are the outer crystals that they can have, because like maybe if you cool it down, it will crystallize and break down all the properties that you need. But it's really far apart. So to be able to make people relate to it, we have to bring in you guys. Like, hey, what do we know? And a lot of time when I see this is when I talk to my supervisors, or to people who are expertise in the field, experts in the field. They have this connections to like, oh yeah, this apply to this. I know this guy who make these particles and this guy in this
Mm.
industry, because that's really give physics legitimacy in the field.
Yeah, like maybe, the methods Feely's developing could be, reconfigured for soft material physics and used in these kind of like drug delivery things one day. And of course there's like a
Mhm.
big gap there, right? But there's always big gaps. Yeah, I completely agree. Convincing people what you're doing is useful is difficult sometimes, especially when you're really deep in theory land like us. Like, the math I'm doing is for a gas that's at 0 Kelvin. 0 Kelvin gases don't exist. Why am I doing it? Right? And it's like, well, you can get gases that are in the nano Kelvin range, and those aren't really like, you don't see those every day on the side of the street. But if you can create those and understand those, then maybe you can use them to understand how quantum computers work, because it's a simpler system than a real life quantum computer would be. And then once you understand that, you can build a quantum computer, and then from that, once you got a quantum computer, you can do all kinds of really big things and make a lot of money. So it's like, yeah, you kind of have to follow this chain of thought depending who you're talking to, I guess.
I think we have that in common of how do we what's the word I'm trying to look for here? What's the scenario I'm trying to look for here of like how do we contextualize the problem, I guess, into a way that is both simple for us to process and simple to explain, right? Like as a from a in an engineering lab, we make a lot of assumptions on the biology that just are just they're so heinous. Like an actual biologist would look at all of the way that we simplify, say, cancer. Like a cancer biologist would hate us if they saw, what we were talking about behind closed doors. As if, cancer is just this one simple thing. And it's just like, oh, all cancer are the same. It's like, no, no, no. There is like so much diversity from person to person, from cancer to cancer, et cetera, et cetera. But as an engineer, you're just like, oh, well, all cancers have these big spaces that are like 100 nanometers big. So I guess just make 100 nanometer nanoparticle and you're fine. And it's like, well, no, that's not really it. But to solve this problem, we're going to start here and then we're going to see what other problems can we solve using that same thing.
Well, physicists would say cancer is a homogeneous sphere with uniform density of one.
Yeah.
So I want to get back to you. So what got you interested? I'm walking back in time. What got you interested in science, like growing up? Like what was your story? Like some people like Liam is a late bloomer who really got into like science in like high school and such. Some people, grew up, let's say their parents really, instilled the love in science into them. But so what's your story? What got you into science? Because, it is a commitment to do a PhD and such. And
Mhm.
I'm sure you have some story on how you got into at least chemistry and um, biology, and when did you run away from physics?
Oh, just got to slide that in there, eh? Of when did you? So I think there's two main ones about why. There's two main moments that I think of why I started liking science, then a little bit more chemistry, and then there's one of physics that's pretty obvious, and I think most people will probably relate on this one. But the first one was when I was a kid. All of my little general science questions that I had, like why is the outside of my glass of water, why is that foggy and whatever? Like my parents could not answer them for the life of them. Love them to death. My parents are the furthest thing from science people. They do not understand the simplest thing, like this a single thing about science. Love them for it. They're very much like arts, more on the arts side. They're very talented in the arts. So, me being stubborn, I needed to know those answers and I wasn't going to take, oh, don't worry about it as an answer. So, I just kind of was always interested in learning those things that maybe weren't as obvious or as talked about in our family circles. That was kind of like the very first one at a pretty young age. And then once kind of got into around high school age, I found that one, I was pretty good at it. Like I was interested and I would do well on tests and whatnot. And then also, even just a little bit before high school, like I had a lot of my family members had cancer. So I knew that I had, I had this interest in, okay, well, how can I make them better? Like, is there something that I can do to learn about it and then try and make it better? And so kind of melding these ideas of, wanting to help people, wanting to help my family, and being generally interested in this counterplay or this interplay between chemistry and biology was, okay, I really want to take this a step forward and I want to try and make a difference in whatever way that I can, and whatever way that I think is best suited to my talents. And it just kind of, I guess, happened to be this chemical biology and like biomedical engineering avenue. Now, where I ran away from physics was pretty early in high school because I sucked at it and I hated it. And mostly I hated it because I sucked at it. I think it's very cool when it's explained at a very high level, like the way that you guys do. But when I have to do math, oh my God, the number of mistakes. I can barely, guys, I can barely add. Like, I can barely add. So when it started to get into physics and, all of all of the cool things you hear in, high level physics, when it all comes down to math, I knew it wasn't going to be for me. I was like, you know what? Kinematics in grade 11 is enough for me. See you guys later.
I mean, I did the same thing for biology. I learned text when I took a taxonomy class. Like, I'm out of here. I can't memorize all this stuff.
Oh,
Like,
that's
I don't want to
bad.
do this. Like, this is torture.
Yeah, you heard me earlier. I couldn't even say half of those things. Like, how am I going to understand them?
Angiogenic.
Yeah, yeah, yeah.
Yeah.
Um,
Taxonomy is a tough one though. That'll put you to sleep. Sorry, taxonomists. But
so I think we have enough time for maybe two or three more questions. I'm going to say two for now because the three of us, me, Patrick, and Feely, we all know what the last question will be because we ask everyone the same last question. So you'll hear that. But my question I have is that you do social media stuff. So I don't want to say you're an influencer, but I guess you are. I don't know how it works. You've got an Instagram, you got TikTok. I saw you made some YouTube videos the last couple weeks or 4 weeks or so. So you're getting into that now, and you've got like a decent following. So like, what kind of? I want to know like your experiences there. What got you into that, and how has that been going? And have you learned anything?
oh, the learning never stops. But yeah, thank you for bringing that up. In terms of being an influencer, I think people assume I'm more of an influencer than I am. I kind of just I make them because I have fun. But it really started from the fact that I felt like I was learning a lot of very cool things during my PhD that was still going over people's heads when I would say we'd have these Thanksgiving dinners with my family, and there was still just this communication gap between what I was doing and what I was able to explain. I could do it at a pretty high level of, oh, imagine you have cancer and you want to develop a drug that has no side effects. That's more or less what I'm working on, is try to make these little materials that make you have no side effects. But there were a lot of specific concepts that you learn along the way, as I'm sure you're all familiar with, in your own research. You're like, oh, I kind of know a lot about this side part of my project that I didn't know about before. And I just kind of wanted an avenue to display that in like more layman's terms and more like casual terms. And so I thought social media would be the best way of doing it. And then when I was, you know, floating through Instagram and TikTok, just there was no one doing any biomedical engineering content, at least not in the way that, not covering the topics that I was interested in, and wasn't explaining it in a way that I would have wanted to explain it. So I just thought, well, why don't I just do it? Why don't I just make these videos that can maybe get people more interested and more engaged in these topics that I think are really, really interesting? And other people might, they just don't know it's interesting yet. They don't know it's a thing that people are working on. So let me bring this research to a more, general, non-technical audience. And so that was kind of the goal. And I think when you combine wanting to do that with the fact that I'm very much like a drama nerd, like way deep down, as in did theater camp growing up a couple of years in the summers because my parents needed to be my brother to do stuff. It was just kind of like a match made in heaven, if you will, of, oh, this is very fun, and I think people could get a lot out of it, and I could get a lot out of it too. So
I mean,
yeah.
I think that the best content is the projection of what you're most interested in, right? Like, I think that's like what internet or like what media is made for, is put your passion out there. Like,
Mhm.
it is doesn't matter like what it is. Like, we are surprised like somebody is listening, right? Like, it sounds so random. It just gets some random people. In a sense, they're not like, the most famous scientists in the world or something, but I think they're interested in this kind of genuine passion. So I think before I get to the last question, I want to ask more a little bit on like biochemistry or chemical biology stuff. Like, when do you know when do you become aware of like the mixture of the two? Because like let's say in high school, but biology and chemistry seems like it's a separate subject on the paper, right? So for someone who wants to get into like your field, or a kid to say, hey, like I'm not aware of this. Like what would be the method to become more aware of this type of interdisciplinary or multidisciplinary field?
Well, you can start by following at Maddie C PhD on TikTok, Instagram, and YouTube, and then from there, I think you just kind of have to let it flow through you. I think it's taking the things that people are learning in elementary school and high school and trying to think beyond what is presented and trying to think, oh, how does this apply to other areas? And then, maybe even if it's just for like 5 minutes on your way home from school, you're just like, what is, this topic that I learned today? How does it relate to something else that I'm kind of interested, right? And then just kind of going from there and kind of, I would recommend just always like challenging yourself and looking for new things to enjoy and blending these different concepts together in a way that is, explained simply. Please don't go and read scientific papers. You don't need to do that. If you're like 14 years old, please do not hit up Google Scholar. But even just like a little Wikipedia article real quick or whatever, even getting ChatGPT to be like, hey, what is this what is this concept and how does this general thing work? Anything to really continue to spark your interest in different things would be my recommendation.
That's part of the reason we do this podcast. Actually, it's kind of forces us to talk about things that aren't our immediate areas of research, which is always good because you get like different perspectives, and it's very easy to get stuck in one hole. You know.
Yeah, and I appreciate it. I was listening to the what was that? What was that episode? Was it Ashenov? No, what was that? Oh, I'm forgetting
Was it one
now.
of
I think it was
ours?
you did it back in. Yeah, it was one of yours. It was back in November, I
Oh, the
think.
Aronoff bone,
Um,
maybe.
yes, that's the one. And
Oh,
I was like, I don't know what any of this means. I've never thought about this in a day in my life, but this is sick.
that was a rough one. I don't think I did it justice, but I tried.
I don't know. Maybe it was rough if you're like an expert in that, but as someone who had just heard about this for the first time ever, I thought it was amazing.
Incredible.
And at the end of the day, labels like biology, chemistry, they're just labels, right? Like our interest is pretty much a spectrum. Like we are interested in multiple things, and sometimes biology, sometimes fall into physics. Well, we are getting to the last question we always ask our guest, and it's a loaded question. And as you know, so what are your hopes and dreams now that you finish your PhD? So you got a job. Do you see yourself becoming like the super salesman, or the super scientist, or the scientist that? Do you still want to work in that delivery? Do you see yourself working in this, or you see yourself go back to teaching, or be a professor? Like where do who do you see yourself being in this life?
Yeah, great. Always my favorite question on here. I did not prepare enough for what I wanted to answer with, but I will go with definitely want to still stay in, the biomedical engineering space. I think it is something that I genuinely have a passion for. I would like to go more into the communication route eventually. And something I want to continue to make this content and try and, you know, broaden this field to people who maybe didn't know that they would like this field of drug delivery and biomedical engineering and whatnot. And I think if I can do that in a way that is highly technical and working mostly alongside experts, that would be pretty cool. If it was less technical and more just for a general audience, that would also be very, very cool. So I'm pretty open. I think as long as it is helpful and useful, but also enjoyable is I think where I hope it can be. And I just want to help people in whatever way that I can.
All right, perfect.
Well, thank you very much for coming, Matt. It was great to have you and have these awesome conversations.
Yes, it's great. Thank you so much, guys. I really appreciate it.
So, before we end off the episode with a story and say goodbye, Patrick's going to tell everyone how they can reach us. And you already told everybody, Matt, but maybe he'll do a reminder about how they can reach you.
You know I will. But before we do that, I just want to let you know how you can reach us, whether you have questions about the episode or other episodes in the show, or if you just want to reach out, you want to say hi, or if you want to suggest topics for future shows. You can reach us through email. We are hyperthesis.podcast@gmail.com. You can also send us a DM on Instagram. We are only on Instagram at the Hyperthesis, but you can follow our Instagram page. I don't actually know the name of it. To get updates about when we post episodes, and you can also see pictures relating to our guest topics, including ideally pictures of our guests and something related to what they do. So it's always a good place to get updated on the Hyperthesis and to contact us as well.
The Instagram's called Hyperthesis Podcast. Please go follow it.
Yeah, and it's at the Hyperthesis if you use the at tag. If you're listening to us, congratulations, you found us. Very well done. We are based out of Spotify, but we are on pretty much whatever podcasting platform you may use, including YouTube Music, Apple Podcast, Audible. So if you're listening to some audiobooks and then want to switch over to the Hyperthesis, feel free to do that. Now, if you do have questions for our guest, Matt, what's the best way to contact you? You posted a whole bunch of socials, but what's your preferred method?
Yeah, definitely the easiest way to get a hold of me would be on Instagram, and you can find me at MattyC PhD. So that's MattyC
Perfect. And those are all linked below. So go ahead and find those different links. I have your YouTube, TikTok, and Instagram down there. So go ahead and message Matt with any questions that you might have. Or if you want to send us an email, we can pass any messages or questions along. So feel free to contact us, especially if you would like to be a guest on the show and you're an expert in your field. We would love to have you on and really pick your brain without crossing any barriers, we'll say.
Nice.
Yes, so I will try and keep this quite quick, but based on the topics that we've been discussing today, I was wondering, what's the history of small things? So we spent a lot of time talking about nanoparticles and how they can be used to deliver drugs, but we haven't always known about nanoparticles, but we've still been using them for quite a long time. So the idea of things that are smaller than what the eye can see has existed for millennia, with one of the first recorded explanations of small things coming from the Greek philosopher Democritus. Democritus, who was alive in the 400s and 300s BC, is well known for his introduction of the atomic theory, although I will say that's even a little bit controversial because his mentor is also attributed to it. And his mentor's name was Leucippus. Now, those ideas are heavily entangled, but they describe this small, indestructible structure that exists at some point as you break things down further and further. Now, the word atom or atomus comes from the Greek word for indestructible, and from the explanations that we interpret from Democritus and his mentor, hypothesize that there's an infinite amount of these small particles that have an infinite number of shapes, and they move freely through a vacuum. So he proposed that there were these small particles and there's a vacuum, and that's all that needed to exist. Really not too far off. Now, when atoms met in this vacuum, when they collided, they would form materials, and the properties of these materials were dependent on the composition from these atoms. So humans were made from differently shaped and sized atoms than that of a tree or the wind or stars or the earth itself. Now, these ideas that were expressed were really not far from the truth at all, and it's really quite amazing to see how well those held up. Of course, now we have quantum mechanics and everything, but it took humans a long time to discover both atoms, learn that they are actually divisible, which we've gotten into many times before, and how to actually build structures out of individual atoms themselves. So this will be a very brief history of how we went from thinking about, okay, what is everything made of, to actually using it and just how small we've gotten. Now, while small things are around all of us, including making up ourselves, we've been able to actually use small structures for much longer than you may think. So going back to the time of Democritus, his hypothesis that there is a size in which something cannot be broken down further was in response to an idea that things were created from nothing. And so he said, nope, things are not created from nothing. There's just this infinite number of atoms that exists. However, humans have been creating tiny particles like what he was talking about for millennia. And a common example of this is when making pottery and glasses from ancient artisans. So by grinding down different materials into powders, such as gold and silver, nanoparticles are created, which can then be added to clays. And then when they're fired, their properties come out. And there's some beautiful examples of color-changing glass from the Greek periods and even from Mesopotamia and other ancient civilizations where they've used nanoparticles and their properties for art, for the most part. Now, a lot of these tiny structures and identifying them rely on microscopy and nanotechnology. You can't just look at a nanoparticle. You have to have tools to be able to see it, and I imagine tools to insert drugs into it as well. There's a lot of technicalities, and while we can grind down materials, to actually see them requires going even beyond visible light. So we'll skip over a few centuries between the invention of microscopes and actually being able to see these very small particles, because we could see microparticles, I'll call them at this point, such as cells or viruses even, or other types of microscale structures. Now, the idea of nanoparticles first came about in 1959 from a talk by Richard Feynman, who seemed to just be in absolutely everything physics related, where he discussed the use of precise machines to construct devices down to the molecular level or even the atomic level. Now, this essentially kicked off the field of nanotechnology, which that term actually wasn't used until 15 years later, where it was coined by Norio Taniguchi. But it was separated into two main categories. There is top-up nanotechnology and top-down nanotechnology and bottom-up. Now, keep in mind that the advancements in the idea of nanotechnology was actually really quite recent, so only within the past 60-ish years. But at this point, they were closer to the discovery of the neutron and proton than to modern day. So just some things to think about when these ideas were happening. So the top-down approach harkens back to the days of ancient civilizations where materials are broken down. So similar to how things are grinded down and used in glass and pottery, this top-down approach is able to take a material and then subtract from it, such as what you would see with machining, but incredibly precise with new technologies and processes. One of these processes is called photolithography, which makes this type of manufacturing infinitely more precise than it was with just kind of etching away at things with hands. Now, in photolithography, which has been around for semiconductor fabrication since 1957, it focuses on the use of light to essentially help wear away at certain materials. Now, the term was first coined in 1958 when researchers from the Army Research Laboratory in the United States used the process of light-triggered solubility to create an integrated circuit. So what happens is you shine a light, especially UV light, onto the surface of a material that then makes it soluble in certain solutions, whereas everything else isn't, and then you're able to kind of wear away at it and change its shape. Now, this type of technology is used to create transistors. So the first one was a germanium transistor, but since then it's being used a lot more to create insanely small transistors that follow Moore's law, meaning that today's transistors exist that are smaller than 2 nanometers. So IBM recently announced a 2 nanometer architecture for transistors, which is, very, very small. Now, compared to the first microchips, which contained thousands of transistors in the late 1970s late 1960s and early 1970s, modern commercial chips have an order of 100 billion transistors. And so if you have an Apple M2 Ultra computer, you have 134 billion transistors on board at a 5 nanometer architecture. And some NVIDIA GPUs have almost double that in terms of transistors. So lots of small things. So we're kind of bordering on what the smallest things that we've actually made are. And it's quite recent, and there's a lot of research going into that. On the other side of manufacturing, for the bottom-up approach, it builds nanostructures in an atom-by-atom or molecule-by-molecule basis, some of which we've discussed today, and it can be done through physical or chemical methods, such as self-assembly using certain types of reactions, or through some other methods where you're able to nudge these components into certain types of shapes. I'll just focus on one example quickly, which is a form of carbon, which can form carbon-60 or carbon-70 balls, which are also known as fullerenes, when graphite is evaporated, and then that evaporated graphite is placed into a high-speed inert gas like helium. And I think the term I saw in a methods paper was hypersonic helium, which sounds really cool, but also really hot. Anyway, when cooled and ionized, it forms these balls, and they naturally actually occur in interplanetary gases and soot, and have a diameter of less than 1 nanometer in size. So instead of etching away and trying to chisel away at a material, you can get a lot smaller by trying to construct materials like these balls, which are known as Buckminster fullerenes or buckyballs. They're definitely smaller than transistors, but they really aren't the smallest thing that people have made so far. I do just want to add a quick word about quantum dots, because their name suggests that they are quite small, but despite their name, they're actually larger than buckyballs. They range from about 5 to 50 nanometers in size, but they're still produced for the most part using a bottom-up approach through a variety of chemical and physical methods. And they are something that's being more and more mass-produced over time as they become more common, especially in display technologies. But going back to non-manufactured and non-mass manufactured items, we can turn back to science and its discoveries for the smallest thing that we've created. Now, while I wouldn't delve into the history of microscopy, that's a story for another day, the smallest object created by a person requires and actually uses microscopes, but is a piece of another type of microscope. So the smallest object, according to the Guinness Book of World Records, that we have directly shaped are the tips of a scanning tunneling electron microscope, or an STM. So this structure is incredibly small and consists of a single atom at its point, and underneath that, in its second layer, is 3 atoms, and then underneath that is 7 atoms. So this world record was set in January 1990 by Eigler and Schweitzer of IBM, where they were able to use this very, very sharp tip to move single atoms of xenon on a nickel surface to spell out the letters IBM. Now, STMs are commonly used today. Go to your local physics department or chemistry department. Chances are you'll be able to find them. And we kind of talked about their use along with the requirement for these super, super fine points in our episode with Catherine Boisvert, who does atomic force microscopy. So there are devices running all the time that are creating and smashing elementary particles, but those don't really count as devices that we can use. I wouldn't get into the particle physics, and because we're kind of creating them, but it's also random. So I would say Guinness Book of World Records is correct that the smallest object that we can manufacture is this head, and I think that will stand for a long time. Now, I hope to be proved wrong one day, where we have something that we're able to manufacture smaller, probably out of some exotic material. Hopefully no one tries to recreate a neutron star, because that might get messy, but it would be very cool to see this smallest object and its record be beat. But this was in 1990, so it might not happen soon, but I do hope it happens.
Oh, just quick note. I saw a little seminar talk on petahertz logic gates, and they're sending lasers into these crystals and basically make it act like a C naught gate or something. I don't remember the specifics, but it might be coming. So even if we don't get smaller, we may be able to get faster.
Well, thank you very much for that, Patrick, and Feely as well at the end. And thanks again for coming, Matt. It was great to have you.
Hopefully I can be back soon. Thanks, everyone.
All right, and thanks everybody for listening. We'll see you later.
All right, take care.
Bye, everyone.